Bienvenidos a los seminarios internacionales de fronteras de ciencia de los materiales. Hoy vamos a abordar un tema un poco distinto a los habituales en estos seminarios y es algo muy relacionado con el propio ser humano. Si uno piensa la cantidad de neuronas que dedicamos al proceso de visión, nos podemos quedar realmente sorprendidos porque prácticamente el 70% de nuestro cerebro está involucrado en procesos relacionados con la visión, el análisis de imagen y la utilización de estos datos. Si partimos de esta premisa, pues es fácil entender la importancia de los materiales en mejorar, optimizar o dar visión a aquellos que no la tienen. Hoy tenemos con nosotros a Susana Marcos, es del Instituto de Óptica del Consejo Superior de Investigaciones Científicas, en concreto del Laboratorio de Óptica Visual y Biofotónica, y nos va a hablar de las propiedades biomecánicas de la córnea a partir de deformaciones de la córnea. En estos momentos es directora de Large, no sé muy bien lo que quiere decir, no soy capaz de traducirlo.
Bueno, miembro del Consejo de Dirección.
Consejo de Dirección, vale. De la Sociedad de Óptica de Estados Unidos. De 2008 a 2012 ha sido directora del Instituto de Óptica, al cual pertenece. 2005 y 2006 fue profesora senior e investigación senior en este mismo instituto, y su vida realmente empezó ligada a este instituto en 1999, como podéis ver. Anteriormente había realizado su tesis en física en la Universidad de Salamanca, centro de reconocido prestigio dentro de la óptica en España y en el ámbito internacional, y posteriormente, en los años 97 a 99, pues tuvo una beca Fulbright para una estancia en la Universidad de Harvard, que también, como sabéis, es unos sitios de referencia internacionales. Sus líneas de investigación y... A ver, se me ha ido esto, y trabajo se centran en la óptica visual, en la imagen, en el Ocular Biomechanics y en New Ocular Corrections. Como podéis ver, es un campo apasionante, extraordinariamente interesante, en el cual se hibrida la física, los materiales y la anatomía, la biología del ojo. Así que yo no os robo más tiempo, os dejo ya con Susana. Muchas gracias por estar aquí, bienvenida, y el tiempo es tuyo.
Muchas gracias. Muchas gracias, José Luis. Le he preguntado a José Luis cuál sería el mejor idioma de la charla, y me ha dicho que no importa. En vista de la grabación, pues creo que hablar en inglés a vosotros probablemente nos importe, y a lo mejor a la difusión del evento ayuda. So it's really a pleasure to be here. I really appreciate the invitation from José Luis, also from Fram, who also had the opportunity to visit us. And I will be talking about corneal biomechanics, and we will be thinking about the cornea as a biomaterial, although for us it's mostly an optical element. And I will be using imaging, and these are techniques that we developed in our lab. So we use imaging to understand the cornea, to understand the ocular elements of the eye, and to put it in better context, as I said, the eye is an optical system. The eye projects images of the outside world onto the retina, and we're interested in the optical structure and geometry of the cornea. So this shape of the cornea actually leads to the optical properties of the cornea, what we mean by optical aberration. So the imperfections of the optical system of the eye are to a large extent produced by irregularities or the shape of the cornea, and also the crystalline lens, but I won't be talking about the crystalline lens today. So this optical structure determines the quality of the images that are projected on the retina. But of course, vision doesn't end there. This is just the first step into the optical process. However, the quality of the images projected on the retina have a large impact on the visual quality. We are ultimately interested in improving the visual function of a patient, right? Someone that had a problem in the cornea and that the cornea is replaced, or the cornea is corrected by a contact lens. We are affecting the geometry of the structures, and that has an impact on vision. And clinicians and optometrists and visual scientists in general, their goal is to improve visual function. But this we can only achieve by manipulating the optical structures. And today I will be talking about the cornea, and I will be giving also some examples on how we can make use of the understanding of the corneal biomechanical response to improve vision. Our lab has produced several techniques that allow quantitative imaging of the eye. So what you're looking at there is images that are obtained with a system that we developed in our lab, which is optical coherence tomography, provided with quantitative tools so that we can produce, for the first time, fully quantitative images of the eye. So basically, it's not only like nice images, all the dimensions that you see there are quantitative. And what you see there is a quantitative image of the cornea. As I said, I'm not addressing today the crystalline lens or intraocular lenses, but that's another major field in our laboratory. And what you see here is also the crystalline lens. And this is actually a merged picture with the same image in an eye after cataract surgery with an intraocular lens. So the green lens that you see there is the implanted lens in a cataract surgery procedure. And all this is quantitative so that we know where exactly that intraocular lens landed after surgery, how tilted it is. And this is a really powerful information to aid cataract surgery as well. But as I said, I will be using this kind of technology to understand the cornea better. So I've been talking about the cornea and as an optical system. But today, in the talk today, I will be focusing on, in fact, the eye, and in particular the cornea as a biomechanical system. We talked about the cornea, which is like this lens in front of the eye. Then there is the iris, there is a crystalline lens, and these project images on the retina. Well, not only the cornea is a biomechanical element in the eye. The crystalline lens, of course, is a tremendous biomechanical element because it can reshape to focus near and far. And it's actually changes in these mechanical properties of the crystalline lens that lead to what is called presbyopia, which is the loss of ability to focus near and far, and that results into the need of near glasses or other corrective alternatives for presbyopia. Then, of course, we have the ocular muscles. These are biomechanical elements as well. And in fact, the entire eye globe, the sclera, is also a biomechanical element. And in fact, this is linked somehow to the development of myopia. In myopia, the eye elongates more than needed to have a perfect focusing of the image on the retina. And the mechanical properties of the sclera are somehow related to this excessive elongation of the eye. But for the purpose of this talk, I will be focusing on the cornea as a biomechanical element. So of course, and you as material scientists know very well that the cornea or any material mechanical properties actually arise from its structure. So it's important to understand what the cornea is made out of. The cornea is 78% water, it's 15% collagen, and then there is a 7% of non-collagenous properties. Most of the cornea, and this is what I will be focusing on today, is stroma. And stroma is where the collagen structure is located. Of course, corneal structure determines these optical properties, and it's important to understand this collagen, which makes the most of this corneal structure is made out of. Collagen is made out of collagen fibers that are composed by collagen fibrils, and the collagen fibrils are composed by corneal molecules that are triple helices. And And in the cornea, you can think the cornea as a basket. This weaving of the so-called lamella, that are about 200 to 500 lamella that are composed by collagen fibers. They run from limbus to limbus, so from white to white. And they have an orthogonal orientation in the center of the cornea, and then a circular orientation in the periphery of the cornea. And that is really what provides the structure of the cornea as well as its shape. Of course, another very important property of this arrangement of the collagen fiber is the transparency. So we don't want only a supportive structure for the cornea, we actually need the cornea to be transparent for vision to take place. So this arrangement actually guarantees corneal transparency and also determines the corneal shape. So there are some conditions where the corneal shape is compromised, and this is compromised because of this arrangement of the lamella and the collagen fibers failing. As I said, imaging is essential in understanding the mechanical properties of the cornea. At a structural level, there are two major techniques that have been used to understand this collagen fiber orientation. The X-ray scattering that requires really large equipment and has allowed to understand this arrangement in the center, in the periphery of the cornea. And it's actually very interesting. This arrangement varies across species, porcine, bovine, and rabbit, which are species that we have investigated, as well as human, have a circumferential fibril orientation. And then another very interesting technique that has allowed to understand this fibril interweaving is second harmonic generation microscopy. This microscopy modality actually targets the collagen fibers so that it's possible to measure the arrangement just looking at the collagen fibers themselves. For example, it's interesting, there is a higher interweaving in the anterior cornea as opposed to the posterior cornea. And that's actually something that surgeons know very well. When they're manipulating the cornea, the anterior cornea is stiffer than the posterior cornea. As I said, there's emerging imaging modalities that have allowed us to actually understand the mechanical structure of the cornea from a microstructure point of view. As I said, corneal shape determines optical quality. The failures in achieving an appropriate shape turns out into astigmatism or coma, which is a higher order aberration that produces distorted images. If we have a regular cornea with an aspheric shape, we will have a sharp image on the retina. However, if we have this type of distorted corneas, the images on the retina will be highly degraded optically, and that will result in a decrease of visual quality. There is in particular a pathology that is relatively frequent. It's called keratoconus, that results from a local weakening in the cornea and a loss of collagen fiber orientation. However, the etiology of keratoconus is not well known, but this is a disease that results in a progressive outbulging of the cornea, and it forms a cone. So that's why it's called keratoconus. So the cornea is very compromised. There's emerging, and we will talk about them in a minute, techniques to try to stop the progression of keratoconus, but many of these patients actually end up requiring a corneal transplant. So understanding keratoconus and finding ways to stop the progression of keratoconus is important, and keratoconus is truly a biomechanical problem in the cornea, and of course results in very poor vision. There is an emerging treatment for keratoconus which is called cross-linking, and perhaps the term cross-linking is familiar to you in another area, for example in polymer science. Well, this is something that is becoming very highly used, particularly in Europe, is running FDA trials in the United States. And as in any cross-linking for polymers, is a photochemical process that is induced. In this case, in the case of the these clinical trials, is a riboflavin as a photosensitizer, and then UV light. So it's a treatment that is activated by light, and there is a photoinitiator that is riboflavin. We're actually involved now in another studies in collaboration with Irene Kotivar's lab in Wellman Center in Harvard, where the photosensitizer is another compound. It's a rose bengal, and it's excited by green light. But it's basically a photochemical process that appears to induce the formation of collagen bonds between collagen fibrils. And that will eventually lead to a stiffening of the cornea tissue, and therefore stopping the progression of keratoconus. This is a very crude illustration of what cross-linking does. These are excised corneas from a porcine eye. It's taken out of the eye. Number one would be a non-treated cornea. Number two would be a cornea just treated with a photosensitizer, but not UV light. And then three, receiving the entire treatment. So what you see just very qualitatively is the third, I mean the treated cornea with riboflavin and UV light is stiff, and the other ones have really lost stiffness. There's several treatments that the cornea can receive, primarily to treat refractive errors. Some of them are probably very familiar to you, are LASIK and PRK. So these are techniques where the cornea is reshaped by the use of a laser. It's basically like a sculpt. In a sense, there are some mechanical responses that are important. When you are reshaping the cornea, you are leaving a thinner cornea, and the intraocular pressure can produce a bulging of the cornea if the cornea is left too thin. So LASIK and PRK, very popular treatments for refractive errors, and biomechanical response potentially being important. There is another treatment that can be used for treatment of refractive errors, but it's primarily used now in several countries to also produce some or provide some support to a keratoconic cornea, which are intrastromal ring segments. These consist on normally PMMA segments of a triangular shape or hexagonal shape that are implanted in the periphery of the cornea, and that they produce like kind of a scaffolding to the cornea, and also flattened the central area of the cornea or make it more regular. And this is why they're used in keratoconus treatment, just to try to make the cornea flattened. However, the surgical outcomes are very difficult to predict, and as you may imagine, it's highly dependent on the mechanical response of the cornea. So these areas, and particularly the second one, are areas where a good understanding of the mechanical properties of the cornea is going to be very helpful, because it's going to help us predicting the outcomes, or will help us to really guide the treatment. For example, try to select which are the proper dimensions of this ring to be implanted to produce the desired effect on the cornea. So just going more specifically into corneal biomechanics, the mechanical response of the cornea depends on the collagen structure, of course, and also the cellular matrix. We can analyze the static properties. Elasticity would be the immediate reformation response to stress, or hyperelasticity if we go into large deformations, and also dynamic properties where we look at the time dependency of these parameters. Viscoelasticity, that is thought to believe to result primarily from molecular rearrangement. This is simple mechanics for you. We use the Young modulus in static testing and also a time dependency term for dynamic testing. Of course, we do stress strain, very standard test for Young modulus retrieval, and also we can compute or estimate the relaxation time constant and retardation time constant of the cornea by looking at deformation versus time or force versus time. So how we actually deal with measurements of the biomechanical response for the cornea? Well, most of the measurements so far in the literature are ex vivo, and they depend on geometrical measurements of deformation. So the most standard, and until very recently, the only available data of the corneal mechanical parameters were from one-dimensional extensiometry. Okay, so something like that. We take a strip of the cornea, and then we do stress strain measurements in extensiometry measurements. More recently, and we have contributed to some of these techniques, two-dimensional extensiometry has been used, either mounting the cornea in an artificial chamber and then increasing intraocular pressure and looking at the deformation of the cornea. This can also be done with the entire eye, where the cornea is not cut, but is attached to the limbus of the sclera. And also there is another technique that we developed where we mount flaps of the cornea into a chamber like this, and we increase intraocular pressure. So this way we are looking at different layers of the cornea. To measure deformation, we use imaging techniques so that they can provide us images like this one, images of the shape, or we can also use laser ray tracing, by which we can look at the changes in curvature of the flap, for example. This is just an example of our inflation models where either the cornea or the whole eye are mounted in a chamber with a temperature and humidity sensor and a pressure transducer that allows us monitoring and changing the intraocular pressure within the eye. There is a pressure system, which is basically a pumping needle motorized that allows increasing the intraocular pressure or decreasing the intraocular pressure. And we can do, like an increased intraocular pressure from physiological conditions into a much higher pressure, and then come down. And simultaneously and synchronized with the capture, we can produce images of the cornea and look at changes in the curvature, changes in the thickness of the cornea, and we can have graphs like this one where we're looking at the changes in thickness of the cornea as a function of the intraocular pressure change. And in this case here, we were looking at corneas that were normal corneas, porcine corneas, and then cross-linked corneas. So basically corneas that apparently had stiffened, and this is very consistent with what we observe in terms of thickness changes, where the stiffer cornea, the treated cornea, changes less than the untreated cornea. And very similar for example, the change in the posterior corneal curvature, where again the treated cornea, stiffer cornea, changes much less than the untreated cornea. You can create stress strain curves and look at the Young modulus, and there are also changes that occur that depend on timing. It's not the same looking at the cornea just excised from the eye than looking at it 24 hours later because of hydration changes. But nevertheless, in either case, we find a steeper function, steeper slope in cross-link eyes, estimating an increase in Young modulus by a factor of 1.58. It's actually very interesting and also quite disturbing that the range of variability of the Young modulus of the cornea in the literature is huge. I mean, it spans more than 2 orders of magnitude. The reasons for this are probably the different stress strain testing methods, also different testing routines, also varying post-mortem times, and varying hydration conditions, and that not only include the timing, but also whether the cornea has been treated in some solutions or preserved in a given solution. So that actually leads to a need for corneal biomechanical properties in vivo. First, because in vitro tissue does not behave the same as the in vivo tissue, and also because really what the clinician needs is to understand the biomechanical properties of a patient and apply a given treatment to one patient. So we really need to know the biomechanical properties at the individual level. For example, we can think on several areas where these in vivo data could be very useful. For example, as an early diagnostic tool for keratoconus, we have talked about this disease where the cornea becomes weaker and weaker and it changes its shape, and that leads into a poor vision. Of course, when this happens, it's normally very late. So if we want an early diagnostic tool, it would be very interesting to have a number, to have a measurement of the mechanical properties and see if they are compromised at an earlier level. Also, as I said, some corneal treatments lack predictability. So it would be very interesting, for example, to see if one particular patient is a good candidate for LASIK surgery or for a corneal treatment. It would be interesting to add to the number of pre-operative measurements that are done on this patient, a measurement that gives us a number for the mechanical properties and see whether that patient is a good candidate or not to a certain surgery. I talked about cross-linking as an emerging treatment for treatment of keratoconus, and it's actually a treatment that modulates the mechanical properties of the eye. So it would be very interesting to evaluate the efficacy of the cross-linking by looking at whether the mechanical properties actually are consistent with a stiffening of the cornea. Well, what can be a good candidate for in vivo measurements of the mechanical properties of the cornea? Well, it would be the combination of quantitative imaging, and remember the very first image I showed you, and our capability to really extract quantitative information of the cornea, and actually do it very quickly. The system I showed you is a dynamic system with ERPAF deformation, and in combining this with mechanical modeling. And that would be a new method, or the method that we propose to retrieve the corneal elastic and viscoelastic parameters of the cornea in vivo. Okay, so let's see how it works. ERPAF corneal deformation is actually something that is used to measure the intraocular pressure of the eye. You may have gone to the ophthalmologist, and one of the measurements that is typically done is a measurement of the intraocular pressure that can be either applanation tonometry or ERPAF tonometry. So the ERPAF in combination with shooting an ERPAF on the cornea is something that is done. We will combine it here with a dynamic imaging of really the deformation of the cornea. So we will be able to characterize the corneal deformation upon an ERPAF, and also understanding the contributing factors of this ERPAF deformation from the sclera, from the corneal geometry, from the intraocular pressure, from the ocular muscles, and from the mechanical properties of the cornea that we try to measure, and retrieve the mechanical parameters of the cornea alone. So there's two ways to do that. There is actually a commercial system that has just been released that targets specifically this combination of ERPAF with imaging. This is based on an imaging modality called Scheinflug imaging. And then another technique that we also have developed in our lab, which is combining this ERPAF with optical coherence tomography. The high speed of either system allows us to obtain a full two-dimensional profiles of the cornea, as you will see, many time points through the corneal deformation events, and those deformations being the input to finite element models. And these are just some images. This is a Scheinflug imaging. You will see more, but this is obtained with this instrument. And this is actually not the profile of the deformation, but the temporal profile of the deformation. So basically taking the apex of the cornea and looking at its deformation with time, which is another analysis that can be made. So as for the high-speed OCT, the OCT allows full biometry and corneal topography from that instrument, and as I showed at the beginning, 3D geometry of the cornea and also the lens. And it actually has a high resolution. It's an interferometric technique, so we have a resolution of 3.4 microns in depth, and also high lateral resolution. We can also image up to the limbus, so we have a large imaging range. We use a mirror to actually image the cornea, and then the mirror has a hole in it through which the ERPAF accesses the cornea. As I said, it's very important to obtain quantitative information from these imaging tools. So there is a lot of image processing and distortion corrections involved into getting from the input data to the output data, which are the geometrical parameters of the cornea. In particularly from this technique, it's important to correct for two distortions that occur. This is a scanning technique, so it's important to correct for the fan distortion produced by the scanner. And also, when you think about all these techniques that image surfaces through refractive surfaces, there is also a refraction, optical distortion, very much like when you have like a pencil seen through a water, right? It's a tilt of the rays. Well, that happens too, and that has to be corrected. But once this is corrected, we have full quantitative information of the cornea. What we did in this system, which is a custom developed system, is to combine that with the ERPAF from a commercial tonometer, from a commercial instrument to measure intraocular pressure. And we actually put it apart and use the ERPAF from it. These are just some specifications on the system, the actual range of 7 millimeters, the acquisition rates that are important, 740 kilohertz for A scans, which are just the image of a point, normally the apex of the cornea. And these are the profiles, what we call B scans, that we have a range of 15 millimeters, this resolution of 0.17 millimeters in lateral resolution, and an acquisition rate of 66 hertz. The ERPAF is 20 milliseconds, and it has a Gaussian profile, and I will give you some details on how we characterize the ERPAF. This is interesting, is the corneal deformation imaging. This is a virgin cornea. This is a cornea after riboflavin, which we see it deforms more. And then after cross-linking, what you see is that interior cornea doesn't reach to the yellow line, which was the deformation in the normal cornea. So with cross-linking, that stiffens the cornea, we find a less deformation, which is consistent to the stiffening produced by cross-linking. The other system is based on Scheinflug imaging. Scheinflug is a technique where a slit of light is projected on the eye. And there is actually a trick plate here, which is the CCD camera, or the image plane, is not perpendicular to the optical axis of the instrument, as it's in any optical instrument, but it's tilted. So what we found with that is images with a high depth of focus. Although they suffer from optical distortion correction, and also the particular geometry of the system produces geometrical distortion. And again, this has to be corrected for that to be quantitatively analyzed. This is an example of the deformation of the cornea, again combining a nerve path with imaging acquisition. In this case, it's a Scheimpflug imaging. It's actually very striking to see how the cornea deforms during this air path event. But this is what happens to your cornea when you go and have an intraocular pressure measure taken. Okay. These are just some other acquisitions, in this case for OCT. Here I'm merging the undeformed cornea to the maximally deformed cornea in 3 cases. This is the same eye before a virgin eye, and then after application of riboflavin, and after application of cross-linking. On the top there are the profiles. Here they're the one dynamic deformation. What you see again after cross-linking, there is a much less deformation. It's very interesting that the procedure also produces a thinning, which is a transitorial, but it's a thinning of the cornea, and still we find less deformation. There are many quantitative deformation parameters that we can obtain from these patterns, either the temporal deformation of the apex of the cornea or a spatial deformation of the cornea. We analyze up to 13 different parameters, but I will be focusing on two of them. The temporal profile, we are looking at the temporal symmetry factor. So basically, the relationship between the time to maximum deformation and then the time after maximum deformation. And then in spacer profile, the peak to peak. So at the maximum deformation, the distance between the 2 shoulders. And we performed experiments to test the contribution of different factors to this corneal deformation as a function of intraocular pressure. This is something that you can do in eyes in vitro. You can run the experiment at different intraocular pressures. Also comparing globes versus buttons to understand what's the role of the sclera. Cross-linking as a good model for having different degrees of stiffening in the cornea, and also comparisons of in vivo and ex vivo. In particular, these are the experiments. We ran experiments ex vivo in porcine eyes as a function of intraocular pressure, cross-linking, and then button versus entire eye. We also performed experiments in vivo and ex vivo in chickens, both virgin and cross-linking. So these were the same animals in vivo, and then after enucleation of the eye. And then in humans, of course, this was not the same samples, but we ran in vivo virgin eyes and keratoconic patients, and also ex vivo virgin eyes from human donors from an eye bank. Again, we use this automatic pressure system to change the intraocular pressure, and this was our setting for the experiments. And the cross-linking treatment, we did the standard treatment, which consists on removing the very thin first layer of the cornea, which is the epithelium, and then applying riboflavin, and then applying UV light. And the treatment takes about an hour. This is the standard clinical treatment. These are examples on porcine eyes, looking at the temporal profile and the spatial profile, how the apex indentation changes with time. These are temporal profiles, and on the right there are spatial profiles. What we see is that the deformation is very sensitive to intraocular pressure. As you may expect, higher intraocular pressure reduces the corneal deformation. Okay. Also, the peak-to-peak changes. It becomes narrower as the intraocular pressure increases. The effect of cross-linking, again, temporal profiles and peak-to-peak distance. So what we found is a smaller deformation after cross-linking, but also there is a change in the temporal profile. It looks like after cross-linking, the curve is more symmetric, and also the deformation starts later than in the virgin corneas or after riboflavin treatment. Also, the peak-to-peak distance, particularly for higher intraocular pressures, is higher, which results in a steeper slope in those curves after cross-linking. When we look at what happens when the sclera is present, in other words, when we measure the cornea attached to an artificial chamber versus when we measure the entire eye, there is a higher and faster deformation in cool globes than in corneal buttons, but in general, the contribution of the sclera is not very important, in keeping with the sclera being much more rigid than the cornea. Another interesting parameter is what happens in vivo versus ex vivo. In vivo, we have the ocular muscles that produce some damping, and what we find is this ramp that occurs in vivo at the end of the deformation event that we don't find in vitro. And that's something that is very easy to just calculate and subtract its contribution. This is just a comparison on the same eyes in Chickens, in virgin or cross-linking. Again, we have like this different at the end of the curves, again, due to the presence of the ocular muscles in vivo, but in general, very similar behavior in vivo and ex vivo on exactly the same eyes. This is also interesting. Keratoconic corneas, remember, they were compromised, they were weakened, and what we observed is that the deformation in the keratoconic corneas is much higher than in normal corneas, as you would expect. Okay, so we have these corneal deformation images. We have been talking about corneal deformation parameters, and there's some corneal deformation parameters that are changed with cross-linking, for example, but we have also seen that intraocular pressure and very likely geometry of the cornea all play a role in this corneal deformation. So if we want to extract the corneal biomechanical properties only, then we have to go to more sophisticated models, finite element models, where we can take into account like all the different parameters and extract, retrieve the mechanical properties. So this is what we do in a 2D axis corneal model geometry, where we use the corneal geometry that we obtain from our images. For the sclera geometry, we use the data from the literature, and the lumbus would be like a transition zone between the two, and then the entire eye is filled by fluid. We use this material model for our cornea. It's a linear viscoelastic model with a viscoelastic component using Prony constants and retardation time constants. And we assume isotropic corneas. Basically, all orientations would have the same modulus, and the Poisson ratio we assume to be 0.49 for the sclera and the cornea. And we also assume this density for the sclera and cornea. There are also some assumptions, large activations being deformated. Initial stresses of the ocular tissues were set according to the intraocular pressure, and we don't consider a temperature dependence of the parameters. A very important input in the model is of course, the air path. Okay? The air path that is actually the one that produces the force into the cornea. And this is a function of time, location, and corneal shape. We use the pressure sensor to characterize the temporal profile of the air path. And you see it lasts about 20 milliseconds, and it has this Gaussian profile. The spatial profile is much more complicated because it really varies at every time point. So we actually developed a fluid dynamic simulation where we consider an airflow And we also consider that the spatial profile depends on the apex indentation and the distance from the apex. So our air path characterization is a mix of experimental and simulated results. This is a simulation in ANSYS of the combination of the air path with the mechanical properties of the cornea, following an optimization process. Here you can compare the results from the calculations and the experimental corneal deformation at the maximum deformation. We have is an iterative estimation of the biomechanical parameters that produce the best predictions and the best match between experiment and calculation. What you see here is the experimental versus simulated data, in particular the effect of IOP. So this is a cross-linked cornea, a peak cornea. These are the retrieved parameters in terms of the Young modulus and the viscoelastic constants. And we see that there is a really good correspondence between the model and the experimental data. And it's also interesting that with the same properties, we are actually able to simulate the performance at every intraocular pressure. Also, the same properties provide very good agreement in the spatial profile curves, as you see there, both the experiment and the simulation as well. We went on and actually did the same thing for in vivo and ex vivo human eyes. So this is the virgin human cornea. Again, yeah, modulus and viscoelastic constants here in the downside part of the slide. And again, a pretty good correspondence ex vivo and in vivo. In vivo, of course, we had to take into account the damping of the ocular muscles. And again, a good correspondence both in the temporal profile and in the spatial profile. We perform a sensitivity analysis to actually understand what are the relative contributions of different parameters into this corneal deformation. As suggested by the experimental studies that I had shown, IOP is a major contributor here. We calculated within the change in the physiological range of each of the parameters, which would be the change in the peak-to-peak distance and the apex indentation. So what we found is that the IOP is one of the major contributors actually to corneal deformation, followed by the Young modulus, but also, of course, stiffness, thickness plays a role. So thinner corneas, of course, we did form more. So it's important to have powerful optimization routines to separate accurately the influence of the mechanical parameters and the other geometrical parameters that we have information about. The only open parameter is intraocular pressure, which any clinical measurement of intraocular pressure at this point actually has assumptions on what the corneal mechanical properties would be. So intraocular pressure for in vivo measurements is probably something that has to be inside the optimization as well. Where can we use these technologies for? Of course, keratoconus is the obvious target. And how can we further advance into this technology? Well, keratoconus is a highly asymmetric condition. It produces asymmetries in the cornea. So it's very likely that you have to move from 2D to 3D measurements and finite element model analysis, requiring therefore more complex models of keratoconus for that being applicable for diagnosis. Something that is actually not future is present. We have already applied finite element modeling of the cornea to increase the predictability of these intraocular corneal ring segments. This is an experimental image with our OCT technology of an eye. This is only half of the cornea. an eye implanted with intraocular rings of a triangular section, and this is the result of our finite element modeling simulation of such a condition. Cross-linking is just starting as a treatment technique. There is a lot of room for improvement, and I think these monitoring technologies will help a lot into improving the cross-linking protocols. And as I said, we're working on UV riboflavin cross-linking as well as green light rose bengal cross-linking. And another technology that we are working on in collaboration with Andy Yoon's lab in Harvard is the use of these finite element simulations on OCT vibrography. In this case, it's not an air puff that produces a corneal deformation, but actually sound waves that produce much finer scale deformation patterns in the cornea. And we're applying these same concepts into the simulation of this technology. So in conclusion, quantitative anterior segment imaging, and we've looked at Scheimpflug and OCT imaging, allows accurate measurements of corneal shape if appropriately corrected for distortions. ERPAF corneal deformation dynamics is influenced by geometry, particularly IOP, but is dominated by the cornea. ERPAF corneal deformation is sensitive to cross-linking and keratoconus, and finite element models allows isolating the corneal biomechanical properties from corneal deformation measurements. I would just like to end up by acknowledging our sources of funding, particularly the Ministerio and the Spanish government, an European research advanced grant, also prior to that a European Young Investigator Award, and also Oculus Ophthalmic Diagnostic has provided us access to their system. And last but not least, I would like to acknowledge my group of research, the Visual Optics and Biophotonics Lab at the Institute of Optics, particularly Sabine Kling, who's been the leading author of many of the studies I presented today, but also Carlos Dornosoro, Daniel Pascual, Pablo Perez Merino, and Nander Pekesi, who have all been involved in the mechanical understanding of the cornea, and also our collaborators in Copernicus University and the Fundación Jiménez Díaz here in Madrid, who are our long-term clinical collaborators and from where we actually have access to patients with these conditions. You can follow us on Twitter, where you will have like very active access to our activities, as well as in our website, where all these publications I showed are there to be downloaded. And thank you again for the invitation, and pleasure to have been here. Thank you.
Thank you very much, Susana. Muchas gracias. We have time for some questions, if you want. Excuse me. We have switch on.
Bueno, no sé si seguir en inglés o podemos hablar en español. Muchas gracias, Susana. Muy interesante y además muy bien explicado, todo muy clarito. Y tenía una duda, bueno, esto más o menos me lo conozco, pero me ha surgido una duda que me interesa. Las propiedades mecánicas de la córnea que habéis visto, ¿hay una gran diferencia entre las direcciones? Es decir, ¿es muy anisótropa o es muy poco anisótropa? Porque algo será.
Es una pregunta muy buena, y además creemos que es algo que es muy dependiente entre especies. Es decir, que lo que podemos, por ejemplo, aprender de nuestros experimentos en cerdo, ¿no? Que es unas córneas que tenemos accesibles, no necesariamente es totalmente trasladable a la córnea humana, pero es muy probable que haya anisotropías. De hecho, en los experimentos de inflado estamos viendo cambios distintos de, por ejemplo, de la curvatura con la presión intraocular en el eje X o en el eje Y. En ese sentido, y de hecho es un experimento que tenemos programado para hacer en la próxima sesión experimental, es con el AirPuff, donde ahora mismo solo estamos caracterizando un meridiano, pues coger uno de nuestros ojos, rotarlo y realmente hacer un análisis 3D, por lo menos en este punto, de la medida y explorar la necesidad de realmente tener que hacer medidas y simulaciones en 3D. Por ejemplo, en un queratocono es claro porque la asimetría, o sea, la deformación se produce de manera muy asimétrica. Entonces, es muy probable que al final pues todo tenga que ser en 3D y tengamos acceso a parámetros en distintos meridianos, ¿no? Y
bueno, y simplemente en la literatura, algún ensayo de tracción uniaxial, ¿no hay información de si es más rígido en un sentido o en otro, en humanos?
Hay alguna medida que se ha hecho cortando strips en dos direcciones. El problema es que como es tan variable en la literatura, toda la información que tenemos de strip extensiometry, la verdad es que no sé si hay una conclusión muy clara de ese tipo de medidas en particular, pero sí, no es extraño que haya diferencias meridionales.
Y eso por terminar ya la última y seguir lo mismo. ¿Y cuál de las dos direcciones sería más importante desde el punto de vista fisiológico? Es decir, ¿el queratocono es típicamente en el vertical, en el eje vertical?
El queratocono es típicamente en sentido vertical, pero eso es muy probable que esté asociado a la propia etiología del queratocono. En el mundo clínico, no sé con qué grado de evidencia, pero para la mayor parte de los clínicos muy convincente, el queratocono está muy asociado a, supongo que no en todo el mundo, y que habrá candidatos bueno, pues más propensos a ello o no, a lo que es el frotarse los ojos. Entonces, ese patrón de frotarse los ojos es probablemente más en horizontal, en vertical que en... Entonces, bueno, hay distintos factores. Uno es esta organización, ¿no?, de las fibras de colágeno, que en humanos, por ejemplo, horizontal y vertical no hay demasiada diferencia, pero sí a 45 grados, por eso nuestro experimento planteado pues lo queremos hacer para distintas orientaciones, incluido, claro, orientaciones oblicuas, ¿no? Y en cualquier caso, todo esto va a tener un efecto sobre astigmatismo, sobre lo que es la propia simetría de la córnea. En el ojo humano, y eso conocemos muy bien porque pues llevamos años midiendo lo que son las aberraciones, ¿no?, las imperfecciones oculares, no hay en los ojos normales no hay una prevalencia del astigmatismo exagerada en una orientación o en otra, aunque sí que, por ejemplo, es algo que varía con la edad. O sea, en pacientes con astigmatismo, el astigmatismo corneal va rotando con la edad, con lo cual este tipo de cosas es interesante, ¿no?, el ligar la geometría con la biomecánica y luego pues ir más allá, ¿no?, y ver cómo eso está sustentado por una microestructura y si hay diferencias meridionales también, ¿no?, en esa estructura.
Muchas gracias.
Pues muchas gracias. No sé si queda alguna otra pregunta. No. Pues muchas gracias a todos, y sobre todo a Susana por esta charla magistral que nos has dado y que nos has abierto los ojos, nunca mejor dicho, a un mundo nuevo y diferente.
Muchísimas gracias a vosotros.
Y esta es tu casa, cuando quieras puedes volver a ella.
Muchas gracias.